like to thank Dr. David Kessler for inviting me again this year. So I did a somewhat similar presentation last year, but mainly focused on how to design a good slide presentation. But this time, I think I would change the topic a little bit and focus on how to do a successful scientific presentation. So let's see. All right. So I think it would be nice to start my presentation with a little bit of self-introduction. As Dr. David Kessler mentioned, I'm currently a resident here, so I'm a second-year clinical pathology resident. And previously, I spent like 4 years in the BTPP PhD program, focusing on canine glomerular disease study biomarkers such as microRNA. And then previously, I was in Taiwan doing a 5-year undergrad DBM program, because that's how DBM like in Taiwan. So I just want to point out that even if you are not a native speaker, you can still be successful in scientific presentation. And so my previous oral presentation experience include 2018, I attended the CVM Research Symposium platform presentation. Also 2017, I got the ACVP annual meeting, got the ASVCP Young Investigator Award for my presentation. 2017 and 2018, I attended the graduate student presentation twice. Once is the 3-minute talk, and the other is the platform presentation. And I think for those, I got first place, second place, and also People's Choice Award. So I think there's something I can share about this topic. Mainly when I talk about scientific presentation, I'm talking about a 15-minute long presentation. Usually you present for like 12 minutes, and you have like 3 minutes for questions. And the outline is like, everybody is familiar with this. You have a title, and you have introduction, objectives, material methods, results, conclusion, and also acknowledgements. And I know that in some workshop or some other presentation, they want to suggest you to use like certain number of slides for a certain section, but I never follow the rule. So you just put whatever you want to say and just do that like depending on the content of each section. So I don't never like restrict myself being, okay, my conclusion can only be 1 page, or my result has to be like 3 to 4 pages. And before I go into each different section, I want to talk about the overall attitude you should have when you present your work. So the first thing is you should be proud of your own work. So thinking about like how much time you spend in the lab by yourself alone, those boring, terrible life. And after all your efforts, this is finally a time you can share your result with other people. So at this time, you should be proud of your own work and present the best of your work. Also, when you're presenting, try to create a sense of excitement, making people think your research is exciting, and try to engage with your audience. That means never stand behind the podium. So for all those past 3 different presentations that I shared with you, none of them was done behind the podium. I always walk out of the podium and just like kind of walk around in the front of the room. By doing that, that's the first step for you to getting an award. So never stand behind the podium. And also, the purpose of the scientific presentation, I think it is also important to know that you are not going to scare people with your research, because I believe that all of your research topic must be like very complex, very complicated. That's why you spend several years in your graduate program to figure it out. But the purpose of this presentation is to make people understand what you're doing. And to do that, you need to make it somewhat simple so that people can understand your research. Also, the next step would be not only they understand your research, but they realize how important your research is, so that appreciate your value, the value of your research. And most importantly, make them remember who you are so that you can get an award in that presentation. And in order to do that, you make sure your presentation is clear enough. So the rule of thumb, making clear, is to put less words on your slide. And I'm not talking about like overall less words per slide, but also talk about you should show them as you talk about them. So don't show everything on the slide already, because people read faster than you talk. So after they read everything, they already lost interest of your talk. So just show them as you talk about it. And then make them visible. So you have to choose the font size that people can see. If it's too small, that people can see it. And different font size, I would say, depends on like how big your screen is. Usually 28 to 32 is enough. But for a room like this, probably 32 is better. And I just realized that, so I usually go to a place when I'm going to do talk like 30 minutes or 45 minutes earlier. So when I came here, I just realized that, oh shoot, this room has like all small screens. So I changed the font size of every slide to make it at least 32 so that people can see. Also need to be consistent. If you choose one type of font and then stick with it. And if choose one type of bullet point, then stick with it. If you have period at the end of every sentence, then you have it for every sentence. If you don't, then don't. So just try to be consistent. Because I'm like very terrible audience. Like I have limited attention span, and I also be very picky when I listen to other people's talk. So when I see a line like this, my eyes will fix to that line and be like, how would you use this font for this sentence but not for the others? And I will completely like not paying attention to what you're talking about, but only look at the line. The next thing is you have last words, so you want to include more pictures or icons. But I would mention that these pictures or icons have to be relevant. So don't put something just because you think it's beautiful or it's like somewhat related to your research, but you're not really using those pictures in your talk. So for example, this is what I found online. When somebody talk about planning a presentation, they somehow put a picture of this here. Like to me, like there's a picture, I know, but like it doesn't really tie to the content itself. Maybe it's like somewhere related, but it's like 100% related. So I really don't have to put it here. I only put picture if I really need it. If I don't, then I take it out. Also, don't say things like, oh, sorry, it may be difficult to see. Because when you show a picture and you're saying that, means that you are underprepared. Like you did not even realize whether people can see your picture or not. You just put it there. And during your presentation, you just realize how embarrassed it is. And you have to say something to cover that embarrassment, being like, oh, sorry, it may be difficult for you to see. So just never say that. And if you want to say something like that, meaning that you can make this picture bigger or you can even take it out. Also, only use high quality pictures. So the only thing that can be pixelated is your cat. Other than that, don't put pixelated picture. And I believe there's nothing better and a good example. So I'm going to share the presentation I did in the ASVCP, the American College of Veterinary Pathology annual meeting, so that you have a better idea of like a complete picture of a scientific presentation. So start with the title. Here's my title. So my research topic is actually very boring. Like, you couldn't win an award because it's so boring. It's like comparison of different methods in order to prepare biofluids in dogs for small RNA sequencing. Like, it sounds very boring. But how can I make it more interesting? So I would like to break down different factors of this title slide. So the first thing is the title. The title usually has to be the same as your abstract, and even it would be the same for your future publication. Also, personally, I like to include some pictures or icons that are relevant to my topic. So in this example, I have a red circle with a syringe in the middle. That means blood. And I also have like kidney and bladder in a yellow circle. That means urine. Because I'm looking at those blood and urine as biofluids in dogs. And I also have a machine that I represent for sequencing. So those are relevant to my topic. And then, let's see. And I also include my advisor and school logo as well, because I think it is important to acknowledge your advisor or your co-authors, and it is also important to show which school you're from. But my personal habit is I won't have them on each slide. I just have them like on the title page and maybe in the acknowledgement. I don't want it to show on every slide. So in order to find a good logo, I think you guys already know this website, but if you don't, just Google TMU space CVN space logo and click on the first result. And on that page, you can find whether you want your logo in maroon color or you want them in white color. So this is just an example. This doesn't mean that those logos are in the maroon square. This means that those logos are white and they're just kind of showing a background so that you know they're white. Those could be helpful. And then move on to the introduction and objective. So I want to show you a normal example of introduction. So for my research, the introduction could be, okay, microRNA, also known as mRNA or miR, are small non-coding RNA, 21 to 25 nucleotide long. And then miR is present in all types of biofluids. And maybe you provide some example, like could be serum, could be urine. Also talk about small RNA sequencing, also known as RNA-seq. It's a powerful tool in characterizing microRNA expression. Small RNA-seq workflow, just like overview, because they don't understand what small RNA-seq is. Then you have to talk about, sorry, I have to show this. So it starts with the RNA isolation, and then library preparation, and sequencing, and analysis. So this is just a common introduction background slide that you will see in every scientific presentation. Like, you know it, you understand it, but now you wonder, how does it tie to my research topic? Like, those are just general background knowledge that you can use in every small RNA sequencing or microRNA sequencing related research. But how does it tie to your research topic? So, and then the normal objective would be, like you should show in one line to say, okay, we would like to compare different RNA isolation methods and different library preparation methods of canine serum and urine for our small RNA sequencing. That's just like very boring. So what's wrong with those traditional presentation? So your audience is basically dead now. They're already lost interest. They don't want to listen to you anymore. And even at the very beginning of your scientific presentation, like nobody is listening right now. And then, even though you want to explain every new vocabulary in the topic of your scientific presentation, you also want to explain it in a way that is relevant to your objective. And also, one thing can help you is if you use some pictures or icons. And I would suggest you to use this strategy. First, there's A, then B, then C, but there's a D. So that's why we are doing this study. And this strategy is not only useful in presentation, but also would be very useful in your grant writing as well. So when you talk about 5 elements of story, because essentially when you do a scientific presentation, it's like you're telling a story to other people. 5 elements of story would start with a setting, so you talk about introduction, background. And then you have character, which would be your experimental subjects or your material methods. And then there must be a conflict. So whatever you tell them about the background has to lead to a conflict, which in our presentation would be a knowledge gap. And knowledge gap will also lead to objective, so that there's a purpose of your study want to fix this knowledge gap. And then the plot would be your hypothesis, your experimental design in order to answer this question, and the resolution would be your results and conclusions. So what I did that presentation is, remember I have small RNA sequencing in the topic. So I started with what is small RNA sequencing. So I say small RNA sequencing is a powerful tool in characterizing microRNA expression. Compared to the traditional method like microarray or QRT-PCR, in those methods you need to use pre-selected microRNAs However, in smRNA sequencing, you don't have to pre-select your microRNA, and this method is even more specific and more sensitive. Then I will talk about, in human, those microRNAs are present in all types of biofluids. So it could be like citrus fluid, could be CSF, could be saliva, could be semen, could be urine, serum and urine. However, currently in veterinary medicine, most of the study in dogs focus on serum and urine. So that ties back to my biofluid that I use in the study, that I use serum and urine. And then the next slide, I will talk a little bit about the smRNA-seq workflow, and I'm doing this for a reason. So a normal smRNA-seq starts with RNA isolation. So you have like a bunch of heterogeneous RNA sequence. And then you perform the library preparation, which you add adapters to both ends of those sequences, and then you reverse transcribe them into cDNA. Later in the sequencing, you have a glass slide, and you have your sequence attached to those slides and have complementary fluorescent signal bind to those slides, bind to those sequence, and then you record your raw data. After the data analysis, you would know which microRNA does the sequence belong to. And I'm saying this because in this workflow, let's see, if it show up, the library preparation is the most challenging part because the most commonly used library preparation kit, the Illumina TruSeq kit, require 200 nanogram per microliter of total RNA as input. Alternatively, you can use 2 microliter of microRNA as input as well. However, according to a recent study, the total RNA present in biofluid is only 14 to 184 picogram per microliter. So that's much smaller than what we need to have for library preparation. And because of that, we face some challenge. In order to fix that challenge, we have like 2 options. One would be to increase our input volume. The other one would be identify viable methods for limited input volume. And the second one is what I'm going to do in the study. So here I just want to show you that in addition to show it in words, you can also include some bar charts to show the difference of the concentration so that people can visualize the difference. And the second part is I don't have a reference slide at the end of my talk, because even if it showed them, the first common thing would be they're too small. Like you have like 20-something reference on the same slide, people cannot see. The second one is even if they can see, they don't know like where these references came from. So I usually show references, I talk about it. Like here in the corner, I have like the author, the journal, the year. That's all I would show in the presentation. Okay. Just cite while you talk about it. Okay, then move on to the material and methods. A traditional material methods will also be like this. Like we have RNA isolation using 2 ml of canine serum, and then we have like 3 different isolation methods for serum. Also use 10 ml of canine urine, we have like 3 different isolation methods. And then library preparation, we have 2 different library preparations. And then we use CPSS 2.0 and DSeq2 for data analysis. This is like pretty standard material method. But what was wrong with this type of presentation? Because your audience just died again. Like they finally have some interest about your objective and what you're going to do, but when you present the material method and then completely lost their interest. So especially for this part, I would say a picture is worth 1,000 words. So this is how I present it. For the method, for RNA isolation, I have 2 ml serum, 10 ml of urine from our dogs. And then for each type of sample, I compare 3 different isolation methods, these 3 for serum, and also these 3 for urine. And then jump to library preparation. Remember we talked about the most commonly used library preparation kit is the Illumina TruSeq. But I also include another kit, the BioNext Flex, which require much less much less total RNA as input for the library preparation. So now people have understanding why you're comparing these 2 different library preparation kits. And then after I'm saying all that, I just want to like remind them what my study is about. So I'm going back to the objective and to re-emphasize. So we have dogs, we have serum, we have urine, and we're trying to compare 6 different RNA isolation methods, also compare 2 different library preparation methods. So I believe by this point, they already remember what your study is about. So when you present the result, they can understand what you're talking about. And then after that, I will use CPSS 2.0 and DSEQ2 for the analysis. So for the results section, also another common problem would be people be presenting tables on slide. I would definitely say table is the worst choice. So this is the table that I eventually put in my publication. But remember, that's publication, that's not presentation. If you present this, then people just be like spend tons of time like reading it by themselves and not listening to you. So in addition, instead of using tables talking about the RNA quantification, I use something like this. So I showed them serum and urine of those 3 different methods, and I also showed them the total RNA concentration. And use some animation, make sure they focus on those methods that gave me the highest 2 RNA concentration, because I have limited budget and I can only choose 2 methods out of 3 going through the library preparation and sequencing. So remember to display data without using tables. And move on to library preparation. So I just changed the topic. And then that also leads to the ultimate results. For one of the method, I was able to compare 2 different library preparation. And I was only be able to use 1 library preparation. And the other one just doesn't work. And also for urine, fortunately, both methods can be used to compare 2 different library preparations. And then, let's see. And also when you show the graph, don't use the graph that you are going to use in your publication. So this one is exactly the same what I put in my paper, but you can see that there are several problems with this figure, is that the legend is too small that people can read. Then also the legend on the right-hand side is kind of complicated, although it separates out serum and urine, but it has combination of RNA isolation method and library preparation methods. So it's kind of complicated. So in my true presentation, I remade all the legends, make them big enough and obvious enough that people can see. And also make the legend bigger so that on the x-axis you have PC1, 51% variance, and the y-axis you have PC2, 70% variance. And also when I talk about it, I have a pet peeve for laser point, so I don't like to use laser point at all. And I feel like if you want to point out something in your talk, that means you already thought about it. And since you thought about it, there must be a way for you to highlight it on the slide already. So as I talk about it, I would say, okay, focus on this is the serum results. And you notice all these are urine results, so they are very different separately on the x-axis because they are different symbols as we expected. And then if we focus on only those urine results, you can see that those symbols are separated by their library preparation methods. So the library preparation true-seq would be on the right-hand side, and the library preparation of bio would be on the left-hand side. And also, when I show the overall result using a Venn diagram, I would talk about the graph and be like, each oval represents the number of microRNA detected in that method comparison. So for example, the combination of using the XOR and EZ isolation method with the bio library preparation would give you 115 microRNAs. And same as this conversation would detect 96 microRNAs in your sample. And I show my comparison as I talk about them, being like, okay, so now when I compare different isolation method with the same library preparation like true-seq, you can see that the combination of the XOR and EZ with the TrueSeq gives you more microRNA. When I compare under BioO, the XORNA is still the better one that gives you more microRNA. And then when you compare different library preparation method using the same RNA isolation method, the BioO is always the better one. So it's better here and also better here. And in the end, I would remind them, if you use the worst combination, then you will probably mix 99 microRNAs that are not detected in this combination. But if you use the best one, the microRNA you missed will only be 29. So this is certainly like the best combination that we found. And then in the end, I talk about those 36 overlapping microRNAs that you will always detect no matter like which combination you use. Those microRNAs covers all these highly abundant microRNA expressed in the canine kidney tissue. So here I'm include the relevant icon so they knows that I'm talking about kidney right now. So what I just did is trying to tell the story and show one thing at a time, and my interpretation will always lead to the next thing I'm going to say when I present the result section. So move on to conclusion. Also, the common type of expression you see in conclusion would be like, okay, first thing, we successfully performed RNA sequencing with adequate total RNA from serum and urine. Second thing would be, okay, the library preparation introduced a higher variation in detecting microRNA in the isolation method. And the third one would be, we identify viable RNA isolation in the library preparation method for limited volume biofluid. So even though those three covers all your conclusions, but the way you present it make it boring, and then even though they're impressed by your questions, your methods, your results, but in the end, they couldn't take much out from this conclusion. Like there's no impression of this conclusion. If they close their eyes, they will not be able to remember anything. So remember I showed this picture in my objective, talking about what I'm going to achieve in this project. In the end, I show a similar graph, being like, here's my conclusion. So I use 2 ml of serum, 10 ml of urine from dogs, identify the best RNA isolation, one for serum, one for urine. Also, in the library preparation, I identify the BioO next flex being the best library preparation method. In doing that, people would have an impression of what you just did, and it is quite vivid because I'm showing using those icons. So summarize your research. You need to remind them of your objectives. And then you prepare some take-home points so that when they close their eyes, they can remember something out from your talk. And then make them think you're awesome, like showing them pretty pictures, pretty icons, make them feel like, oh, you just did awesome research. And the research is just recently published in a journal and made it to the cover of the journal. And I think just because the Venn diagram color matches the theme of the journal, so that's why they selected and put it on the cover. Make it pretty. So that's also very important. And the last part is the acknowledgement. So don't forget to acknowledge your funding agency. And oftentimes, when they talk about those agencies, you can include their icons as well. And then remember to thank your advisor and your lab mates. And I think for this part, it can be like, try to have more fun with it. So this is what I did. So I acknowledged my scholarship to study abroad and also the core facility funding provided by the Texas A&M AgriLife and also the Kelly Cook Endowment for kidney disease research. And then for my lab, I just put on the lab picture and like describes whoever that is. And for this part, I think it is important to not to thank your ex-boyfriends, your husbands, and not putting pictures of the kids, unless it's your boss's kid that I can that still can make it to the acknowledgements. So just make sure it's relevant to your scientific study. So take-home points for today's presentation, keys to successful scientific presentations. First thing would be to always remember to show your best work, be proud of it, and bring the excitement in your presentation. Also, keep your audience alive. Don't kill them in your presentation. Make them engage with them, and then make sure they pay attention to your talk. And then just have fun, because you have 12 minutes. It's the time you shine. Just have fun and be confident and say things out loud, present your research. So after this presentation, I think you guys might still have some questions about, okay, so how can I find good quality pictures or icons? How can I determine using a pretty color theme throughout all my presentation? And how can I make sure every picture and icons on my presentation aligned well with using just pressing one button and not just like dragging those pictures around? So that goes back to the presentation I gave last year. It's called Grad School 101, Design a Great Slide Presentation. And you can find it on YouTube if you type the title. Or right now you can scan the QR code with your cell phone. So I talk everything, how to find good picture, how to find good icons, how to change the color of your icons so that match with your presentation, and then how to align everything on your PowerPoint slide to make them pretty.